So next topic from our syllabus is about oncogene, proto-oncogenes and tumor suppression gene. We'll see one by one. So oncogene, onco means cancer. Gene is gene, we know gene. So oncogene is a gene which is responsible, which has a potential to cause cancer. And it has been observed that in cells, wherever there is cancer or tumor cells, these genes are found mutated or they are expressed at high levels. We know that whenever this life grows, we have got a very fine balance between new cell formation and death of cell. Cell of death, it can be because of most easy example of this, which you are very well familiar about, is RBCs. We know all RBCs, they have their lifetime of around 120 days. So that means whatsoever RBC is found today, within after 120 days, it has to be got destroyed. It will be taken up by the liver. So this is what cell programming is there, and this is, we call it as a programmed cell death, to what apoptosis is called. So this kind of apoptosis is there, that is programmed cell death is there, and cell formation is there. There is a balance. So if the death is more, then we can say that the RBC level will decrease much than normal. It can result into anemia or any other such problems. If it increases, then we'll call it as a polycythemia, or it can be a sort of cancer which is resulting into more cells. So this kind of balance is there, that is maintained by the various genetic makeup, which causes the balance between formation of cells or growth of cells and the death of cells. So when critical functions are altered and malfunctioning, these activated oncogenes can cause those cells designated for apoptosis to survive, and not only proliferate but survive and go beyond it, proliferate it. So you can imagine that maybe RBCs, again we know about it, the RBC which should have died after 120 days, and against it new RBCs are going to be produced as usual. The cell which has to be died should have been destroyed, or there should be apoptosis. This cell is not only remaining there, but fortunately this do not proliferate. They are not developing, RBCs are. But suppose instead of RBC there is another cell. That cell which has to be died, the cell which should have been died, it should be proliferating now. So the cell, there should be count, particularly if that cell was there, such 10 cells should have been died, should have been apoptotized. So if these are not, so it should have been become 0 out of 10 cells, suppose there are. Now what will happen, this 10 cell not only will remain there, instead of becoming 0, this 10 will be there, and now this 10 will cause proliferation, means they will grow. So this is what the reason behind tumor genesis. This most oncogenes, they become as a proto-oncogenes, that is the initial stage of oncogene, and these are involved in the cell growth. These are actually the genes which are involved in the cell growth and proliferation, or they causes inhibition of apoptosis. So the genes which are basically responsible for cell division and cell growth, or those which are the genes which are responsible for inhibiting the apoptosis, they are caused as a oncogenes, because they are responsible for either stoppage of cell death, or which is a programmed cell death. The apoptosis was a programmed cell death. So if any genetic material which is stopping this, which is changing the sequence of this programmed cell death, either these agents or those which are causing growth or proliferation of the cells, these are all type of genes will be coming under this. Usually multiple oncogenes along with mutated apoptotic or tumor suppression gene will act to make the cancer, and many oncogenes has been identified in human cancer. Many cancer drugs that are targeting proteins, they are encoded by oncogenes. So this is a new way of medicines is been grown. If you remember, we have seen that MABs, that is monoclonal antibodies, then NIBs, that is example imatinib, mesylate, etc. These are acting on these tyrosine kinases, etc. So these are various places where the drugs are acting, and these are new sets of various drugs which are there. So we can have this simple figure which is showing over here, that is there are many agents which can have potential to cause cancer because of their chemical structure. There are many such environmental there, including many chemicals are there. Then this chemical or it can be sunlight, it can be some other light. So anything, it can be any physicochemical agent which have property or which have ability to act on the genes which are there. So there are proto-oncogenes which are not oncogenes, but they can be converted easily into oncogenes by small mutation. Now this mutation, we know there are various types of mutation. Mutation can be brought because of either physicochemical types of agents. It can be light, it can be radiation, it can be anything, it can be any chemicals. There are many such chemicals are there. So these proto-oncogenes will be mutated and they will converted into oncogenes. They get activated, and because of that activation they will lead for a new cell which is not required, which was not at all required, and this is we call as a cancer cell. So this is how these genes are working. So first of that, we have seen that it is a proto-oncogenes, it is a normal gene. This can become a oncogene due to mutations or increase in the expression. They code for the proteins that helps to regulate the cell growth and differentiation. Proto-oncogenes, example of these proteins, you know, there are many proteins we have seen which are taking part in replication when various phases of cell cycle. We have seen all these proteins. Okay, so all these proteins, it can be this kind of proteins, can enzymes or any kind of this restriction point drugs, these agents which are activating. So by this mechanism they will regulate the cell growth and they will regulate the differentiation. They are often involved in the signal transduction and execution of mitogenic signals. You have seen all these mitogenic signal or MAPKs, mitogen activated protein kinases, and because of these protein products which are there, because of this MAPK signals, these proteins which are there, activated protein kinases are causing activation of these proteins. These protein products can become a tumor inducing agents and it becomes oncogene. Example of proto-oncogenes are known as RAS, WNT, MIC, ERK and TRK. These are various genes which are there. This MIC gene is implicated in Burkitt's lymphoma, and it starts when there is a chromosomal translocation. We have seen chromosomal translocation. We have seen that is in the pair of gene, some portion of the previous gene is exchanged with other. We call it as a translocation. Okay, in insertion only one portion goes to other is a insertion, but when there is a exchange, we call it as a translocation. So these are RAS, WNT, MIC, ERK, TRK. MIC is in Burkitt lymphoma, and there is a chromosomal translocation. It moves as an enhancer sequence, so it enhances the sequence of RNAs within the vicinity of MIC gene, and it codes for widely used transcription factors. We have seen that there are variety of transcription factors, and these are very, very important for the expression of gene. So this enhancer sequence is wrongly placed. This transcription factor are produced at much higher rates, and because of that more transcription is there, and because of that more such proteins and enzymes which are required for replication, cellular protein, etc., they all will be activated. Another example of oncogene is BCR-ABL gene is very popular, which is found on Philadelphia chromosome. It is a piece of genetical material which is seen in a chronic myelogenous leukemia, which is caused by translocation of pieces from chromosome number 9 and 22. BCR-ABL code for a tyrosine kinase which is constitutively active, and it leads to uncontrolled proliferation. So these are many such agents are there, many such examples are there. So what are the steps into that? It becomes oncogene by a small modification in the original function, and these are three basic steps or activation. One is mutation within proto-oncogene that will cause a regulatory region. Then it can be because of or it can lead to increase in the protein activity. It can cause loss of regulation. Then second thing is there can be an increase in the amount of certain proteins, that is increase in the protein concentration, and it is caused by increase in the protein expression through misregulation, increase in the protein that is mRNA stability. So it will prolong the existence. We know that after the work is there, immediately we have got enzymes which will cause lysis of this RNA, but if it is not done, this enzyme is not properly produced, then this mRNA will stay for a longer period. So instead of one cycle of protein synthesis, it may go for more than one cycle. So more proteins will be synthesized, and this will lead to one after another activities. Then there will be gene duplication, one or other type of chromosome abnormality. We have seen chromosomal abnormalities, aneuploidy, and many such chromosomal abnormalities we have seen. So this can lead to gene duplication. That will result into increased amount of protein in the cell, and again this will lead for cancer-like activity. Then chromosomal translocation, we know about it, what it is, and it causes the exchange of genetic material between two chromosomes, and this leads to fusion between proto-oncogene and second gene, which creates fusion protein, and there is an increase in the cancer or oncogenic activity. So these are various situations. Will not go for all details. There are so many examples of these are there. The expression of oncogenes can be regulated by microRNAs. These are small RNAs having 21 to 25 nucleotides in length, and they control the gene expression by down regulation of the gene regulation. Mutation in such microRNAs, they are called as oncomedes. Onco is cancer, and MIRS, that is microRNAs. So it leads to activation of oncogenes. Further, antisense messengers RNAs, they could also be used to block the effect of these oncogenes. So there are various ways by which we can classify oncogenes, and not exact any standard. Example, if you know, in pharmacology we can classify the drug on the basis of mechanism of action. We can classify on the basis of chemical classes. So similarly, we can have variety of classification system over here. Sometimes they are grouped by spatial way. Spatial means starting from outside of the cell to coming up to the nucleus or chromosomes. So this is space wise, from one area to other area, so outside the cell, or on the receptor present on the cell membrane, then inside the cytoplasm, or organelles which are present in that, then nucleus and various parts, like that we are coming. This is space wise. Then chronologically, that is paralleling the normal process of the cell signal transduction. Okay, one after another, what happens to next, what happened to the next, what happened to the next. So these are various categories which are commonly used, and how it is there. Example are this is growth factors or mitogens. We know that they are act through various protein kinases, insulin like growth factor, MAPK, etc. Example of this is CCs, and they are implicated in various glioblastomas, fibrosarcomas, osteosarcomas, breast carcinomas, melanomas, because they can induce all kind of cell proliferation. Then other are receptor tyrosine kinases. There are many growth factors into that, that is known as epidermal growth factors. Receptors are there, that is EGFR, platelet derived growth factor receptor, vascular endothelial growth factor receptors, and HER2 are there. HER2 is quite popular, particularly since it has been very much known about breast cancer, gastrointestinal tumors, non-small cell lung cancer, and pancreatic cancer. In all these are involved. So what they are doing is they transduce the signal for cell growth and differentiation. Means when there should not be any signal. Normally in MAPKs or this tyrosine kinases, when these receptors are stimulated, they are because of certain stress, or because of certain, because we call them as a SAPKs, stress activated protein kinases. So in certain such circumstances, these are these ligands or these biochemicals are secreted. Um, but in these cases, without any such provocation, these are coming, and they are continuously forcing for cell growth and differentiation. Then cytoplasmic tyrosine kinases. Example of these are SRC family, 6-ZAP70 family, BTK family of tyrosine kinases. Just now we have seen Philadelphia chromosome, etc. They are known for colorectal and breast cancer, melanomas, ovarian cancers, gastric cancer, head and neck cancer, pancreatic cancer, lung cancer, brain cancer, and blood cancer. Almost all type of cancers, these tyrosine kinases are implicated, and therefore many drugs, these NIBs I told you, they are the inhibitors of tyrosine kinases. We shall be seeing them when we shall be seeing cancers. They are mediating the response to and the activation of receptors of cell proliferation, migration, differentiation, and survival. Then next are the cytoplasmic serine threonine kinases. These serine threonine kinases are important in apoptosis. Okay, so if this apoptosis is not taking place properly, that means there will be they are also called as a caspases. Serine threonine kinases are also known as a caspases, and so they are working through RAP kinases, cyclin dependent kinases. We have seen CDKs during replication. They are responsible for malignant melanoma, papillary thyroid cancer, colorectal cancer, ovarian cancer, etc. So they are involved in the organism development, cell cycle regulation, proliferation, cell survival, and apoptosis. Then other are GTPases and transcription factors. There are many such. GTPases example are working through RAS proteins. They are responsible for adenocarcinomas of the pancreas and colon, thyroid tumors, myeloid leukemia. They are involved in the signaling a major pathway leading to cell proliferation. And lastly, transcription factors, they are through MYC gene and responsible for various lymphomas, leukemias, breast cancer, pancreatic cancer, retinoblastoma, and small cell lung cancer. They regulate transcription of genes that induce proliferation. So these are actually the agents. How this product are there, how these things are working. We know growth factors are there. There are multiple growth. We have just seen few names, but there are many growth factors which are either cell specific. If you remember GMCSF, etc. If you remember the formation of various blood cells. So we have found that granulocyte, macrocyte, or granulocyte macrophage colony stimulating factors. So like that, many growth factors. These colony stimulating factors are also growth factors. Then there are insulin like growth factors. There are many growth. We have seen few of them just now. So these are all growth factors are there. These all growth factors are responsible for the stimulation of machinery for proliferation and growth. And it can be either specialized or non-specialized cell. They are secreting it. They can induce cell proliferation either themselves or nearby cell or distant cell. Anywhere they can do it. And oncogene may cause a cell to secrete growth factors, even though it does not normally do so. So oncogene may get expressed and may cause synthesis of a growth factor. That growth factor will go and act on one or other cell and cause proliferation. So this will initiate the cycle, and that can it can continue for the cancer. It will be induced its own uncontrolled proliferation, that is autocrine loop, means it will go on synthesizing itself. So these are the activities. Then other are receptor tyrosine kinases. They will add phosphate groups to other proteins, and when the phosphate groups are added to other protein, then we call them as a protein kinases. Means proteins are activated. Because of this activation of the protein, they can turn them off or turn off So turning on means this protein will be taking part, or they will be activated to play some important role like enzyme or any other such important job, and turning off means they will not able to do the function. They will add the phosphate groups to the receptor protein at the surface, and this will receive the protein signals from outside the cell and transmit to the inside of cell. So there are such many tyrosine kinases, they will be working and they are responsible. And lastly, these are the RAS proteins, we call them, we have just now seen. These are small GTPases. We know GTP, that is guanosine triphosphate, that hydrolyzes GTP into GDP and phosphate. This RAS is activated by growth factor signaling like EGF or TGF beta. That is, TGF is a transforming growth factor, EGP is endothelial growth factors, and acting like a binary switch. Binary means both way, making on or off, and it causes growth signaling pathways. There are downstream effectors of RAP. They include 3 mitogen activated protein kinases. We have seen all these pathways. So we have seen these pathways, MAPK pathways, then tyrosine kinase pathways. So all these pathways are playing important role. RAF and MAP kinase pathway, MAPK. This is important. There are 4 varieties of MAPKs: MAPK, MAPKKK. That is kinase. That is 3 Ks are there. There can be 2 Ks. MAPKK, that is MAP kinase. So these are various kinases. That is example of this MAP kinase is MEK. Which was MEK was an old name. If you remember, all this we have seen earlier when we have seen MAPK pathways and ERKs. So these are various ERK mediated mechanisms, and they will regulate genes, they will cause the cell proliferation. So thus these are various oncogenes. These are oncogenes which are playing important role into forming cancer. These are basically 2 ways, I repeat. These are what either they inhibit the apoptosis, that is programmed cell death, or they will cause or they will initiate the cell growth and cell proliferation. So any of these simply is simple thing is what is either they will decrease cell death or increase the proliferation, and because of that the number of cells will increase. This number of increase in the number of cell, we call it as a cancer. So there are this variety of mechanisms it is having, and these all mechanisms are resulting into cancers. So there are variety of reasons the cancer can be. And now that's what important in this is, now because of all this advanced technology and advanced knowledge about genetics and what is happening, we are now comparatively better placed to have better drug in cancer. Up to this, in last 8-10 years, there is a drastic change is taking place in the anti-cancer treatment. Earlier only whatsoever things were there that were very common, that is very few drugs were there into it, acting on one or other place, and they all were particularly in destruction of cell, and therefore they are very toxic because they were not able to distinguish between the normal cell and cancer cells. So when cancer cells were supposed to be destroyed, even normal cells were destroyed, and therefore very, very high adverse effects these drugs were showing. Fortunately now it has been this knowledge about this genetics and the involvement of various genes or various genetic material, maybe transcription factor or various kind of proteins which are regulating it. This is helping to understand what are the precise mechanism happening at genetic level. So instead of destroying it, can we prevent this overformation, and which can be more safer than having anti-cancer treatment. So this way it is going on, and maybe if the work goes on continuously, it will help into more such drugs. So these are how various drugs are. Will go for the opposite to this, that we call it as a tumor suppression gene. So we have seen the previous one that these were oncogenes. Oncogenes were the genes which were responsible for initiating either growth or proliferation or inhibition of this particular activity of apoptosis. Now we have here is a tumor suppressor gene, as it shows, its name shows, it will suppress the formation of tumor, and therefore we can call it as an anti-oncogene, because oncogene was responsible for development, growth, proliferation. So this will regulate the cell during cell division and replication. If the cell grows uncontrollably, it will result into cancer. But this will possible if the tumor suppressor gene is mutated. It will result into loss or reduction in the function, and along with other some genetic mutation, this could allow the cell to grow abnormally. The loss of function of these genes may be even more significant in the development of human cancer as compared to activation of oncogenes. This is very important. That means the cause of cancer is not that, or in most of the cases, the cause is not more activation of oncogene, but it is rather more failure of tumor suppressor gene. So that means tumor suppressor gene is very important in controlling the cell growth and cell development. And if these are normal, that even oncogenes will not able to do the things in proper way. But if these are not there, then even the normal without oncogenes, whatsoever development will be taken, it will become uncontrolled and unregulated one. So this is very important, that is, they are playing more important role comparatively. They are classified in two categories. It is again not a very official one, but these are based as a three category. One is called as a caretaker gene, another called as a gatekeeper gene, and more recently one more category has been proposed by scientists, is called as a landscaper genes. So what are the jobs? Caretaker genes, they will ensure the stability of genome via DNA repair, and subsequently if when mutated, they will allow mutation to accumulate. Okay, so they will take care of this genetic material, and we will see that this mutation to accumulate and not immediately expressed till it is required. In the meanwhile, it will accumulate, it will not express, but the gatekeeper gene will regulate the cell growth by inhibiting the cell cycle progression, or they may induce the apoptosis, means they will either stop the cell cycle growth, or it will not allow cell cycle to progress ahead, or it may cause apoptosis. And the landscaper genes will regulate growth by contributing to surrounding environment, because we have seen that it is not only genes but whatsoever surrounding them. We have started this knowledge including nucleosome and presence of histones, histone proteins, histone acetylator, deacetylator. You can imagine everybody is playing a role. Example, histone acetylation is responsible for easily removal of genetic material from histone, so that it can be processed for replication or expression. So even that level. So this is what environment. So this, it is not directly gene, but this type of all enzymes which are playing important role in each phase or each part of these genetic activities. So these landscaper genes, they will regulating growth by contributing to surrounding environment. Example, simply, if we cause the deacetylation of histone, that means it will again because of deacetylation, it will again bind to nucleosome, that is histone proteins, and it will not be accessible for gene expression. So if this simple change is there, that can control it. That is, once we make the acetylation, deacetylation, then again it will be okay. So this acetylation, deacetylation cycles, if we control, then also it will playing role. So therefore we call them as a landscaper genes. They will contribute to the surrounding environment. When mutated, they can cause environment that promotes unregulated proliferation. So this is not only one example about acetylation and deacetylation. It can be nuclear factor presence. It can be presence of various enzymes which are taking part in each and every step. So anything which is less or more, that will making impact on this. The classification schemes are evolving as because of many techniques in molecular biology, genetics, epigenetics, many techniques are coming. So slowly again it will be added into it. What are the function of these tumor suppressor genes? They are more precisely the proteins they are encoding. Means as a gene do not do anything. Whatsoever genes are doing, they are doing through the proteins they are encoding. That is what we have learned so far. Genes are not directly doing anything. Genes do what genes are getting expressed because of their expression, because of the RNAs which are formed, because of their binding with the ribosome, protein is synthesized, and these proteins are playing role in the cell physiology or anatomy. So here the tumor suppressor gene, through their proteins which they encode, they can have a repressive effect on the regulation of cell cycle. They can promote apoptosis, or they can do both. Tumor suppressor gene proteins, they function in various ways. What are the mechanism? They are acting as a repression of genes. We have seen expression and repression, or stimulation and inhibitions. We have seen various words for that. Various codons which are there, okay, and these codons and various machineries are there during nearby it. So there shall be repression of genes that are essential for continuing the progression of the cell cycle. Example of these are various transcription factors. They are playing role as a either repressor or stimulation. If genes are not expressed, then cell cycle does not continue and effectively inhibiting the cell division. Then second mechanism or second way is coupling of the cell by DNA damage. So as long as there is a damaged DNA in the cell, it should not be divided, and if the damage can be repaired, then cell can continue. So this is a second way by which this can do. They will not allow the repair of faulty gene, and then it will not do the cell division. Then next is apoptosis. If the damage cannot be repaired, the cell should initiate apoptosis and remove the threat it poses to the organism as a whole. So they will bring out this apoptosis. Then cell adhesion, some protein involved in the adhesion prevent tumor cells from dispersing, and they will block loss of contact inhibition, and they will inhibit the metastasis. Metastasis is the most dangerous thing in cancers, which normally occurs in the malignant cancers, that the cancer cell spreads to various organs, and then it becomes very dangerous to control, and almost it is very difficult to save the patient from cancers. So these are proteins, they are known as a metastasis suppressors. These are again helping as anti-cancer. And lastly, DNA repair. This caretaker genes will encode proteins that will function in repairing mutation in the genome. They will preventing cells from replicating, and particularly with mutation, the increased mutation rate from decreased DNA repair leads to increased inactivation of other tumor suppressors and activation of oncogenes. So increased mutation rate from decreased DNA repair. So mutation, if it is more in genes where there is a DNA repair is decreased, then it will lead to more inactivation of tumor suppressor gene, and because of more inactivation of tumor suppression, more cancer chances are there, or more activation of oncogenes is possible. So it is important that DNA repair is done, and then only the mutations mutation should not be there in the where DNA repair has not been taken place. So very important examples of these are various proteins. Example is retinoblastoma protein, that is called as a PRB. It was the first tumor suppressor protein which are known, and it is preventing the cell cycle progression from G1 phase to S phase. It binds to ETF group of proteins, and it represses the necessary gene transcription. So this is PRB was the protein. Then P53 is very, very popular one. It is present in many, or it is implicated in many cancers and many places. It encodes the protein P53, which is nicknamed as a guardian of the genome. Very, very useful, very, very important. So it has many different functions in the cell, including DNA repair, then induction of apoptosis, transcription and regulating the cell cycle. If the p53 is mutated, it is involved in many human cancers. Around 6.5 million cancer, 37% are because of the p53 mutations. So you can understand how important it is. Then this makes a popular target for new cancer therapy. Many drugs are actually being tried towards this p53 protein. This homozygous loss, we have seen homozygous, and LOH we have seen heterozygous loss of heterozygosity. So here it is homozygous loss of p53 is found in 65% of the colon cancer, 30 to 50% of the breast cancer, and 50% of the lung cancer. That indicates how important p53 in cancer, or how much involvement this mutation of p53 is playing in cancers. This mutated p53 is also involved in the pathophysiology of leukemias, lymphomas, sarcomas, various tumors, and this p53 can be inherited in various other syndromes also. That is like Lee-Fraumeni syndrome, where other cancers can be there in patients with having this syndrome. Means they are very much prone for cancers. Then another very popular protein gene product is BCL2. It is the family of proteins that are involved in either inducing or inhibiting apoptosis, and the main function is involved in maintenance or composition of the mitochondria membrane. It prevents the cytochrome C release into the cytosol, and when cytochrome C is released from mitochondria, it starts to signaling cascade which cause apoptosis. Again, this is very important mechanism in apoptosis, that that is mitochondria gives signal of death. When mitochondria says that I don't want to make energy more, then it will release this, and accordingly the apoptosis will be there. Other are SWI, SNF. These are various chromatin remodeling complex, and they are lost in around 20% of the tumors. The complex consists of around 10 to 15 subunits which are encoded by 20 different genes. Mutations in the individual complex can lead to misfolding. Misfolding means genes are folded, they are coiled. If it is not properly coiled, then it will be misfolding, and this is having ability to complex the work together as a whole. It is ability to move the nucleosome, and it contains the DNA, allowing for transcription or block transcription from occurring in certain genes. So mutating this ability could cause genes to be turned off or on at a wrong times. So where it should not be on, it will become on, or where it should not become off, or when it should be on, it will not on, or where it should be off, it will not off, like that. So these are four basic problems it can make, and because of that it will result into cancer. So these are the various reasons. There are many proteins which are known, but these above few are very prominent one, and they are responsible for various cancers. So I hope you understood this. These are the important genes. You can have these details available over there. This will play important role in various cancers. So this is what about various cancer, how they are, how the genes are responsible for cancer, and what are two different types of opposing gene. One are oncogenes and proto-oncogenes category, and another are tumor suppressor genes. So when there is an imbalance between these two, then there will be either hyperactivation of oncogene or lesser activation of this tumor suppressor gene. Then this will be reason for many of the cancers. And as I told you, nowadays scientists are trying to target this type of mechanisms, so that this will be more safer than killing the cells, because cells, all the type of cells getting killed, and that can be very bigger adverse effects. So I hope you are understanding it. If you have any difficulties, you may put on our group, and take care.